I'm Vladislav Sandler, co-founder and CEO of Hemogenyx. Today we begin a series of informal videos where we will try to explain how we make our discoveries, who are the people behind these discoveries, and what is the goal of our work. A few words about the origins of Hemogenyx. Hemogenyx was co-founded by myself and Alexis Sandler in 2013. In 2014, in the end of 2014, we participated in the biggest in the world business competition, which is called 43 North, and we, together with a handful of other companies, became finalists of this competition and ultimately won against about 7,000 of companies all over the world. To develop a cure for acute myeloid leukemia, we developed a novel bispecific antibody that redirects immune cells of the patient to find and kill acute myeloid leukemia cells in the same patient. In parallel to that, we decided to develop a cell-based approach to fight leukemia. Recently, we announced, for example, a development of novel chimeric antigen receptor, which is based on our proprietary antibody that we use to construct our bispecific CDX product. To test our new bispecific antibodies, our CDX product candidate, we had to develop a new type of humanized mice that would have human immune system. These mice proved to be better than anything known to us existing on the market today, and development of these mice allowed us to form a collaboration with Johnson Pharmaceuticals. This company asked us to build a new, non-existent today model of human lupus using our mice. So we signed this collaboration, we went into work building this model. While building a human lupus model using our proprietary mice, we decided to look closer at the origins and the mechanisms of autoimmune diseases. And without going into much details, we were able to develop a new approach to treat lupus as one of the autoimmune diseases, and in general we believe that we have a new method of treatment of pretty much every known autoimmune disease today. Since this is an introductory video, I'm not going to take too much of your time explaining minute details of different projects that we're running. Instead, I will try to show you what the company looks like inside, what type of instruments we're using, and who are the people behind our discoveries. Let's go for a tour. This is one of the labs that we use, and some of the instruments. You can see our famous robot, we call her Sophie. You can see also a few instruments such as RT-PCR. This is our tank where we keep our cells. It's liquid nitrogen, it's filled with liquid nitrogen. And you can also see our small kitchen where we make coffee while we have a break. You can see one of our scientists, Karina Sirachinsky, acquiring data from one of the experiments. We are located in the incubator, in the biotechnology incubator of the SUNY Downstate. This is State University of New York, Downstate Division. There are a few companies located in this incubator, including Hemogenyx.
Hi, I'm Ronan Ben-Jehuda, and I'm a scientist at Hemogenyx since January 2019. Before joining the company, I did my PhD in the Technion in Israel, studying the electrophysiological abnormalities of induced pluripotent stem cell-derived cardiomyocytes that were generated from patients carrying PRKAG2 mutations or suffering from Duchenne muscular dystrophy. Additionally, using CRISPR-Cas9, my research showed that all abnormalities were eliminated after correcting the mutation. Here at Hemogenyx, I take part in developing both cellular and murine model for autoimmune diseases such as lupus. Additionally, using single-cell sequencing, we're aiming to develop novel targets for various autoimmune diseases and to explore new opportunities for developing and producing neutralizing antibodies for infectious diseases.
My name is Karina Zurchinsky, and I've been a scientist at Hemogenyx for over 2 years. Prior to joining the company, I studied chemistry at Macaulay Honors College at Hunter College, where I also gained research experience in bio-nanotechnology. In addition, I was an Amgen Scholar at Columbia University, where I got research experience in biomedical engineering. I joined Hemogenyx because I saw it as a good way to blend my knowledge and experience in stem cell and cancer biology. To that end, at Hemogenyx, I work on developing mouse models of human immune systems and diseased human immune systems, like in cancer and leukemia and autoimmune diseases like lupus. We use these novel mouse models to test drugs and to find novel targets for these diseases.
Hello, my name is Raymond Liang. I'm a scientist at Hemogenyx. I've been here for about over a year. I did my training at Mount Sinai in New York, where I completed my PhD. There I studied hematopoietic stem cells and also more downstream processes, including erythropoiesis, which is the formation of red blood cells. Currently at Hemogenyx, I'm working on improving our humanized mouse models to study various blood diseases, and I'm also developing a cell therapy.
Hi, I'm Elena Shrestha. I'm a scientist at Hemogenyx. I received my PhD from NYU School of Medicine in cell and molecular biology, where my research primarily focused on dissecting the molecular mechanisms by which diabetes and hyperglycemia exacerbates the development of cardiovascular diseases, in particular atherosclerosis. At Hemogenyx, our team has developed a highly specific and effective treatment for acute myeloid leukemia. It's an antibody-based therapy that basically utilizes body's own immune system and immune cells to find, attack, and kill cancer cells. And my role in this is to basically design and implement experiments to test the efficacy of the molecule in preclinical setting, in cell culture, as well as in animal models.